As we have seen in our previous module about water and the energy source in the fermentation media, but in this module we will talk about in detail the carbon sources and what are the factors that affect on the choice of the carbon source. Because the carbon is very important part of the fermentation media, that major part of the fermentation media after the water is carbon source. So there are different factors that affect on the choice of the carbon source. That first is the rate at which the carbon source is metabolized. That directly influence on the formation of the biomass as well as on the production of primary and secondary metabolites. As we know that there are different ranges of our fermentation product. First one is the biomass, second is the metabolite that can be a primary metabolite or that can be a secondary or tertiary metabolite. So as concerned the fast growth sometimes that lead to sometimes in a positive way, but sometimes the fast growth of any organism that can affect on the product in a negative way. Sometimes that can lead to, especially in case of secondary metabolites, when there will be a more growth, then there will be a less productivity of the secondary metabolites. So that's why we have to be very careful while selecting the carbon source. So as concerned, when the main product, the fermentation, that effect on the choice of the carbon source, that is basically that product result from the direct dissimilation. So sometimes that our product is directly related to the carbon source. So in fermentation, such as ethanol and single cell proteins, then the production where the raw material contribute 60 to 77% of the total production cost, and the selling price of the product will be determined by largely on the cost of the carbon source. In case when we use the molasses, that what is the price of the molasses, then the product costs vary as the cost of the molasses vary. So that's mostly the cost contributing factor is the carbon source. So as concerned, when we have the selection of the carbon, then we should know about the media preparation, particularly the sterilization, that effect upon the suitability of the carbohydrate for the fermentation process. So it is very much critical while having the sterilization process in different carbon compounds. As an example, when we utilize the glucose as a carbon source, then there is the need of separate sterilization without that may react with the ammonium, and if we utilize with that ammonium, that then there will be to form a black nitrogen containing compound which partially inhibit the growth of many microorganisms. Sometimes when we have a sterilization of glucose like carbohydrates at 121 degrees Celsius by autoclaving process, then there is a choice of polymerization. So that become the unavailability of those carbohydrates for the fermentation process. In case of the starch and such carbohydrates, when we use the polysaccharides, starch and then CMC, carboxymethyl cellulose, when we use and then we make in a sterilization process, then they gelatinized, and then there is a viscous, giving rise to a very viscous solution that can affect on agitation and so many that, even when there is a concentration more than 2%. So the choice of the substrate in a carbon that sometimes is directly affected by the government legislation. Sometimes you are allowed to use those carbon sources, or sometimes you cannot use those carbon. What is meant by that? Suppose if we want to utilize those carbon sources which are already in is utilized by the human food or as a part of animal feed, then there is a laws by the government that you cannot utilize for industrial fermentation, because due to the risk of the utilization. The purity of the carbon sources that also affect on the choice of the substrate. So sometimes, as an example, there are some metallic ions that must be removed through those carbon sources. In case of the citric acid production, when there is a metallic compounds are present in that effect on the citric acid production, because the citric acid production process and such kind of other product that is very sensitive to the enzymatic effect of the organisms. So when we use those compounds which have the high metallic concentration such as molasses, then it is very difficult to control the system. So we have either removed those metallic concentration. In case of the molasses, we have to clarify the molasses just to remove the debris and those metallic compound before using the molasses into such kind of fermentation process.